Hello, everyone. Welcome to today's live broadcast, Predictive Gene Signatures, Molecular Markers Distinguishing Colon, Adenomatous Polyp, and Carcinoma. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by AB SciEx. I am Judy O'Rourke, and I'll be your moderator for today's event. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Janice Drew, PhD. Dr. Drew is a Senior Research Fellow, MSc Human Nutrition, Course Coordinator for Molecular Nutrition in the Rowick Institute of Nutrition and Health within the College of Life Sciences and Medicine at the University of Aberdeen. I will now turn it over to Dr. Drew for her presentation.
Okay, so today I'd like to present some of the work from my lab on the development approaches that we've used to produce predictive gene signatures using multiplexed gene expression profiling. The advent of microarray technologies has led to a massive increase in and illuminated a new molecular complexity in carcinogenesis, identifying aberrant gene signatures associated with initiation and progression. And the focus in my lab has been on distilling the discovery of these broad molecular signatures into smaller gene sets of appropriate density to allow us to develop clinical assays that have potential to identify and measure predictive gene signatures. And this would specifically allow us to develop diagnostic tools to use predictive gene signatures in disease detection and staging and also progression, and also allow us to have an insight into selecting appropriate therapeutic or identifying therapeutic responses, and also selecting treatments that have efficacy and compliance. This prompted us to explore multiplex gene expression profiling, and we looked at the GenomeLab System. This was specifically because this allowed us the options to multiplex gene expression assays using around 20 to 30 gene targets. And we could also custom design these in-house. And very importantly, when using clinical samples, we were able to use very small amounts of starting material. So hopefully during this webinar, I'll be able to highlight the potential of multiplexed gene expression, developing in-house multiplex gene expression assays, and demonstrate their potential for cancer screening and detection, and also looking at how these may be used in developing drugs, and also the selection of effective interventions for individual patients. And of course, this is becoming increasingly important as we seek to stratify approaches to patients. So the aspects that I will specifically look at are issues around the design of the multiplexes, issues surrounding tissue optimization of the assays. I'll take a look at some of the issues that multiplexing has provoked around total RNA quality. And I'll also take a look at some of the analytical and clinical validation work that we've done with the GenomeLab System gene expression technology platform. I'd then like to finish off by focusing on an application of our in-house custom designed GEXP assays to assess tissue status and pathology. So our first multiplex came about following a macroarray study in which we had tried to identify inflammatory gene targets that were associated with the early events in colon carcinogenesis. And this was conducted using a targeted macroarray of around 100 genes. And this identified 14 gene targets that showed differential expression in adenoma and carcinoma samples when compared to normal tissue. We then set about validating these 14 targets using a single-plexed real-time PCR approach, and this validated the gene expression results from the macroarray study. Our next step was, of course, to then take these gene targets forward into a further clinical validation in which we would attempt to verify that these gene targets were differentially regulated in larger patient sets. But of course, the macroarray and the real-time study was obviously a fairly time-consuming exercise, and we were looking for something that would speed up our processing of tissue samples. And this prompted a further look at the GenomeLab system and the options within that for multiplexed RNA quantitation. And this led to the development of a multiplex assay which incorporated all 14 of these gene targets in a single assay so that we could measure the 14 gene targets simultaneously in a single reaction. And this also allowed us an opportunity to validate the GenomeLab System technology platform against both of the other gene expression technology platforms that we'd used for this study. So what I'd like to do now is just take you through some of the processes that are now routinely employed in my lab in custom designing our gene expression assays. And I'd like to use the hCellMarkerPlex as an example of how we develop this approach. The first step, of course, is to select the gene targets that you wish to multiplex. And in this case, we had already selected our 14 targets from the macroarray and real-time PCR analysis. So we gather the accession numbers for these, and this is all recorded into a master file that we keep a spreadsheet of all our multiplexes. And as is now routine, we incorporate perhaps 2 to 3 reference genes in each of our multiplexes. In this case, we had already used GapDH and B2M for validation of the real-time targets and also in the macroarray, so it made sense to incorporate these targets in the multiplex. The next step is that these the sequences which are associated with the accession numbers are then uploaded into the GenomeLab System software system. And we use the Express Profiler component of this to actually design our gene-specific primers for each of the targets. We then take each of those primers and we will then run them through some sort of search engine such as BLAST, just to make sure that they don't have any off-target or non-target homology that's going to cause problems in the assays. And we also gather information on the structural information of each of the targets, such as intron-exon boundaries, for example, and we try to ensure, where possible, that our primer assays will amplify across the intron-exon boundaries. When we're happy with the primer selections, we then begin to conduct the first step of optimizing the assay. Now this requires, of course, choosing your template RNA. In our case, we've been using colon mucosa, and this slide just simply shows a typical colon mucosal sample. This one actually is an ex vivo cultured colon explant. But from this you can clearly see from the gradated scale here that these are very small tissue samples, and we typically use around 20 to 30 milligrams of tissue for RNA extraction. Following the extraction, we then assess quality using both adjuvant analysis and also nanodrop spectrophotometry. Now, for those of you unfamiliar with the GenomeLab System GEXP assays, I will just go through in the next couple of slides just the principles behind the assays. Now, of course, when we design a new multiplex, the first step is to actually then conduct a singleplex of each of our individual gene-specific primer assays to ensure that we amplify a single fragment of the expected size. So each of our targets is amplified using a gene-specific primer, which is denoted by the purple bar here. And each of these has a universal tag attached, indicated by the red bar. So reverse transcription proceeds with the gene-specific reverse primer. And during the first 1 to 3 cycles of PCR, this proceeds with the gene-specific primers for forward and reverse, and both of these have a universal tag attached. In the subsequent rounds of PCR, amplification is then performed using the universal primers alone, and the forward universal primer has a fluorescent tag attached. So the outcome of this is that we generate fluorescently tagged amplicons specific for our gene target, and these are fluorescently labelled. Once we're satisfied that each of our primer assays produces a single fragment of the expected size, we then begin to optimize the multiplex. So the reverse primers are mixed in a master mix, and each of these will be used to conduct the multiplex reaction. So during this reaction, we then amplify each of our targets simultaneously in a single reaction. And we end up with a mixture of reaction products which are all fluorescently labelled at the 5' end. And each of these primers, each of these gene targets, amplified gene targets, differs in size by around 5 to 7 bases. We then take these amplified fragments and subject them to capillary electrophoresis and fragment analysis using the genetic analysis system. So following capillary electrophoresis, we obtain a readout that looks something like this. This is an electropherogram here. Each of the panels represents a reaction that's been conducted in triplicate, and this is a routine procedure that we process the reactions in triplicate. From these electrophoresed products, you can see from the blue peaks here, each of these represents one of our amplified targets within the multiplex. And the red peaks indicate the position of a size standard that's incorporated with the PCR reaction prior to capillary electrophoresis. And this allows us to then, or allows the software to actually assign sizes to each of these amplified fragments, and you can see that in the electropherogram here. And we obviously then check that each of these is appropriate for the expected sizes within our multiplex. Now the sizes are assigned using a size filter for each multiplex, and from this we can then get a readout of the gene expression levels, which is associated with each peak on the electropherogram. And at this stage we usually export that data into a spreadsheet, and we can then use 1 or 2 or 3 of the potential reference genes to calculate normalization. Now, an important aspect at this stage is to ensure that all of our amplified fragments are within the linear range for the assay. And in the initial stages of optimizing the inflamplex, we actually used a mixture of template, which consisted of a 1:1:1 mix of RNA extracted from normal adenoma and carcinoma tissues. However, when we came to apply this to the individual tissue samples, we did have some further optimization to do because, of course, when you move through the stages of disease initiation and progression, you tend to have quite large changes in gene expression. Indeed, and often in carcinoma, we have gene expression which almost drops out and new genes appear. So in order to capture this linear range, we conducted some further attenuation in each individual tissue sample status. And this is simply performed by titrating reverse primer concentrations in the multiplex mix. And what we're trying to do is ensure that we can identify our targets across the 3 different tissue states and that these are within linear range for relative quantitation. So from this, then, we can output our final multiplex, and this is the inflamplex here, and you can see here on the right-hand side, the right-hand column, the final selected reverse primer concentrations for this multiplex. So once we have the multiplex optimized for the template tissues that we're testing, we can then conduct our multiplex gene expression assays. So the samples that were used in the original macroarray and real-time PCR analysis were now subjected to gene expression profiling using the hMFlamplex, which measured all 14 targets simultaneously in each tissue sample. And from this, we could compile a bar chart here, which simply shows the mean expression levels in each of the normal adenoma and carcinoma samples. Now, when we compared the gene expression data obtained using the hMFlamplex against the gene expression data that we had gathered from the macroarray and real-time PCR analysis, we could clearly see that those gene targets which showed downregulation when assessing using macroarray or real-time PCR also showed a downregulation when measured using the hMFlamplex. Likewise, those genes that showed an upregulation, indicated by the green arrows here, we also obtained an upregulation with the inflamplex. So this was very encouraging, and this prompted us to proceed with a further clinical validation using the inflamplex to assess gene expression in a new patient tissue set. And we obtained these tissues from the Tayside Tissue Bank in Dundee in Scotland. So this table now shows a comparison of the gene expression of our 14 targets measured using the hMFlamplex and comparisons with the previous samples which were collected from the Aberdeen Tissue Bank. So in the table here you can see normal versus adenoma and normal versus carcinoma gene expression. The red coloured boxes indicate a downregulation compared to these, and the green represents an upregulation. And you can see there's a fair degree of convergence across the table, in that when we have genes that were downregulated in the Aberdeen Tissue Bank set, they're usually also downregulated in the Tayside Tissue Bank set, and likewise for those genes that were upregulated. Now, when we this was particularly encouraging because as we proceeded with the clinical validation and we were testing larger patient sets, we were less choosy about selecting samples with the best RNA quality. I should mention that when we conducted the macroarray and real-time PCR study on the Aberdeen Tissue Bank set, we actually selected those tissue samples that gave us good quality RNA. However, the quality of clinical samples is notoriously variable, and you can see here that when we have poor tissue bank samples, poor clinical samples, would tend to have a correspondingly poor quality of RNA, which is shown in the adjuvant traces here on the right. And in the top right, you can see that the ribosomal peaks indicate a fair amount of degradation in this sample. So some of the anomalies between our T-side tissue bank set and the Aberdeen tissue set may partly be due to differences in RNA quality, but also they reflect the heterogeneity of clinical samples and also the heterogeneity in carcinoma. I thought it might be useful to just take a closer look at one of the individual gene targets within the multiplex and examine this more closely. The charts here show gene expression measured in individual patient samples, in normal in dark blue here, normal versus adenoma in blue, and normal versus carcinoma in pink. And you can see in the Aberdeen tissue bank patient set, real-time PCR gene expression in adenoma and carcinoma compared to normal. You can see that the carcinoma typically express a higher degree of variation in gene expression. When we compare the gene expression measured using the hCellMarkerPlex assay on the Aberdeen tissue patient set, you can see that these are broadly similar. In fact, they're very similar. And then when we come to the larger patient set that was tested from the T-side tissue bank, again, you can see similar ranges of expression and variation, and again, this higher level of expression or variation in expression in carcinoma samples. But they're broadly similar, and this gave us some confidence in proceeding with the development of the multiplex assays. We were able to demonstrate that we could obtain comparable gene expression patterns using different gene expression technology platforms, and that the multiplex was performed favorably in comparison with these. We were also able to then apply our multiplex to conduct clinical validation of identified individual targets, and compared these against the initial analysis. And this also highlighted that we were able to obtain gene expression profiles using the multiplex gene expression assay, even when the RNA quality was less than optimal. And during this procedure, we also learned a bit about tissue optimization, RNA quality, and also the sample heterogeneity issues. And I'll say a bit more about those as I proceed. So this gave us confidence then to look at the future potential of the GEXP technology platform to allow us to investigate the function and regulation and characterization of gene expression profiles of using multiple targets in biopsy tissues. So I'd like to, during the remainder of this webinar, focus on the application of multiplex gene expression profiling to identify gene signatures in human clinical samples. Now, as I mentioned earlier, carcinogenesis is associated with aberrant molecular signatures that are associated with disease initiation and progression. And typically, we only have small amounts of these tissues for assay. And as I've already highlighted, they exhibit a broad heterogeneity, and this is contributed by the genetic variation across different individuals. It can be caused by variation in the tissue location from which the surgeon collects the samples. And then added to that, we also have the heterogeneity that is associated with diseased tissue. And in particular, carcinomas can actually have areas of necrotic tissue, and these often yield very poor quality RNA. And we're also aware that clinical samples are often not collected optimally for preserving RNA quality. So taking all these issues into mind, we were still confident that the GenomeLab system would be able to allow us to measure gene expression. And we're also aware, of course, that this broad heterogeneity excludes the use of single biomarkers, and multiplexing then becomes absolutely crucial in identifying predictive gene signatures. The other aspect of the multiplex assays that is very useful is that they're flexible, and by that I mean we can actually custom design, and we can remove or add in new targets as these are discovered or as they become a target of interest. So we can modify these assays as we proceed. So this multiplex, the hCellMarkerPlex assay, attempts to capture that broad heterogeneity, and it incorporates 21 cell markers which mark various cellular processes which include epithelial cell determination. We also have cell differentiation, cell survival and apoptotic markers, and various structural determinants. As is now routine, we include a couple of reference genes, and here we've used B2M and UBCH5B. And we also include the synthetic transcript CAN-R, which is supplied with the GenomeLab System assay kits. And this synthetic transcript simply allows us to measure reaction efficiency during the reverse transcription in PCR. Now, as with our previous studies, we were aware that there was a range of total RNA quality in our clinical samples, and in this study we noted RIN values from as low as 2 up to a RIN value of 10, which is at the high end of the quality scale. So we attempted using linear discriminant analysis to identify a potential cutoff whereby we would obtain or maximize correctly classified tissue samples based on gene expression profiles. And you can see from this chart here that we have this step here at around RIN 5. So we set this as our cutoff for RNA quality. However, you'll note from the y-axis here that even down to the low RIN values, we still have a high percentage of correctly classified tissues based on the gene expression profiles obtained. Okay, so once we had applied the hCellMarkerPlex assay to our normal adenomatous polyp and carcinoma tissue samples, we exported the data, and this was normalized, and then we conducted a principal component analysis. And this simply allows us to look at clustering patterns associated with the gene expression profiles. And you can see here that there were a couple of anomalies, and we appeared to have carcinoma samples that were clustering with the normal tissues in blue here. We then took this up with the pathologists and established that actually one of these was a mislabeling, and that the tumour sample, the carcinoma sample, was actually normal. In the second case, we actually attributed this to the fact that this tissue sample was taken from a very large carcinoma, which exhibited a very high degree in variation in dysplasia. And it may be that the tissue sample that was taken exhibited a more normal profile, or was taken from a more normal area of the carcinoma. And this highlights an important issue in clinical sampling, and we would anticipate that, of course, it might be necessary to conduct multiple sampling of these carcinoma samples with the multiplex multiplexes. So these two data sets were removed, and we ended up with our final principal component analysis biplot that looks something like this. And this simply allows us, as I said, to identify clustering patterns in the tissue. And we can see here that the normal tissues tend to cluster, and the position of each of the genes on the biplot indicate potential classifier genes which are contributing to this clustering pattern. So when we now compile the gene expression data and construct a bar chart showing the mean expression levels, we end up with this graph here, which shows us the mean expression levels in normal adenomatous polyp and carcinoma. And then by conducting an ANOVA, we can identify those genes that are differentially expressed in individual tissues. So the principal component analysis and the statistical analysis of the mean expression data allow us to identify the fact that obviously the pathology plays a significant part in the gene expression patterns observed. But it doesn't establish whether or not we could assign tissue status or tissue pathology based on the gene expression patterns alone. And to do this, we used an orthogonal partial least squares discriminant analysis, and that's shown in the plot on the right here. And this multiple regression analysis allows us to discriminate the gene sets or the gene expression data by maximizing covariance to assign tissue status based on the gene expression pattern or the gene expression signature from the multiplex. From this, we can also compile a variable importance plot, and from this it's possible to identify that collagen 1A1 and SLC9A2 are particularly important in discriminating adenoma and carcinoma. And if we go back to the plot here, you can see that collagen 1A1 is situated up here, and SLC9A2 is at the opposite end. And it's important, of course, to point out here that you can see that the OPLS discriminant analysis in component 1 here, we have a complete separation of the normal tissues indicated by the open triangles here, and in component 2 we separate the adenoma, the grey filled triangles, from the carcinoma, the black triangles in the upper right quadrant. From the rank importance plot, again, we also can see that those genes that are situated towards the left hand of the plot are also some of the important genes that are contributing to the discrimination of each of the tissue types. From this, we can also then produce a matrix which simply established that we were able to correctly identify 100% of the normal tissue samples and correctly identify 88.24 and 94.12% of the adenoma and carcinoma samples respectively. So if we go back to the principal component analysis biplot, this indicates potential classifier genes and highlights those genes that we might want to take forward for further investigation and validation. And there are just a few that I'd like to point out. For example, MS4A12 here is actually a colon-specific epithelial marker. We took this forward for further validation, and you can see in this in-situ hybridisation that this particular transcript has a very specific localization on the surface, the luminal surface epithelium of the colon here, and you can see that by the white staining on the black bright field image here. The in-situ also confirmed, of course, our multiplex gene expression profiling, in that we have a loss of expression of this transcript in the adenoma, and we see a variable pattern in the epithelium in the carcinoma. Now, during our initial gathering of information on the gene targets for the multiplex, we were aware that there were two variants for MS4A12, a long and a short variant. So in order to check that the loss of expression wasn't simply due to a loss of the generation of the long variant, which was the variant that was being amplified in the multiplex, we conducted some singleplex, we designed singleplex primers specific to each of the long and short variants, and then conducted a real-time PCR. And you can see from the charts here, we have normal adenoma and carcinoma levels of expression of MS4A12 long and short variants. And you can see that the gene expression patterns and profiles are very similar for the long and short variant in each of these tissue samples. Another gene target that was highlighted as being of specific interest is LGR5. This, of course, is reported to be a colon stem cell marker. And the colon stem cells are single cells that reside at the base of the colon crypts. And you can see again that the in-situ confirms the aberrant expression that we identified from the multiplex, in that we have an increase in epithelial expression shown in the bright field image here by the white staining over the epithelium. And this is in contrast to the expression in the single cells identified at the base of the crypt. And this highlighted for us the sensitivity of the multiplex assay in detecting this particular transcript from a very complex tissue sample. And finally, I'd just like to point out CDX2. We followed up and validated this particular transcript because it was known that MS4A12 actually has a CDX2 responsive promoter in its 5' region. And we were aware, of course, that we had an increase in CDX2 gene expression in the epithelium in the adenoma at the same time that we had a loss of MS4A12 expression. So CDX2 expression in adenoma, upregulation in adenoma compared to normal, was confirmed by the in-situ hybridisation. You can see that there is an increase in CDX2 expression in the epithelium of the colon crypt here, as opposed to the normal tissue. And we also then followed this up at the protein level, and were also able to confirm an upregulation of the CDX2 protein in the epithelium. So the reasons why MS4A12 expression are lost will require further investigation. So in summary, the GenomeLab System GXP assays have been validated against other gene expression technology platforms and have proved to perform well. This has permitted us to custom design a number of assays and use them in gene regulatory studies. This has included looking at hormonal regulation of the inflammatory targets using our inflamplex, and also designing an assay to look at tissue classification using predictive gene signature assays. And more recently, we've been actually applying the multiplex gene expression assays to conduct human blood gene expression profiling to look at responses to nutritional interventions. And I should also say that we've also been using the multiplex assays to assess drug responses using colon tissue explant cultures. So the custom design options for relative quantitation of multiple gene targets simultaneously within a single reaction using small tissue biopsy samples, and the improved data processing time have presented us with a feasible strategy to measure and identify predictive gene signature assays. And they demonstrate potential for clinical utility as a diagnostic tool to look at disease initiation and progression, and also to look at therapeutic responses. And this is particularly important as we move towards more stratified approaches in medicine. And finally, I'd just like to thank a number of people who've been involved in the development of our multiplex gene expression assays. These include Andrew Farquharson, who's my research assistant and who has conducted most of our GEXP assays. Our genomics unit to maintain and organise all the equipment that we need to run the assays. The statisticians at Biomathematics and Statistics Scotland, Dr. Klaus Meyer and Dr. Graham Horgan. Our collaborators at Dundee University, Professor Robert Steele and Professor Frank Carey, and also the Tayside and Aberdeen tissue banks for supplying us with tissue samples. And I'd also like to thank our funders, of course, the Scottish Government and NHS Grampian.
Thank you, Dr. Drew, for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is a two-parter. First part is, what do you think about the relation between gut microbes and colon carcinogenesis? And the other part is, I also read a scientific paper that talks about using a microbial signature as a biomarker for diagnosis of colon cancer. Did you happen to read it, and what do you think about it? If you need me to read that second part again, after you answer the first one, just let me know.
Hello, I haven't read this paper specifically, and certainly we do have groups working at the Institute here who are looking at the gut microbiota and links to colon cancer. And certainly it should be possible to obtain bacterial samples from colon cancer patients. And I would think it would also be possible then to design multiplex assays to look at changes in gut microbiota. At the moment, some of the studies we're doing here, we're looking at the entire gut. They tend to be, we're still dealing with big data, so we're still characterizing the gut microbiome. But as we sort of focus and target on specific bacteria or specific bacterial gene changes associated with colon cancer, it would then be possible from these large data sets to sub-select, as I've perhaps described, select a smaller gene set that could be used as a kind of signature assay. So I certainly see that that's a strategy that could be developed.
Okay, there appear to be no further questions from the audience, and so we will soon conclude the presentation. Just want to let you know we have a quick poll to answer for this presentation. It'll pop up on your screen in a moment. The poll question is, having observed the application of multiplex gene expression with XP-PCR, are you planning to apply this technology in your laboratory workflow over the next, and then you indicate the time period. Give you a couple of moments to answer that question. And while you're answering that question, I would like to thank our sponsor, AB SciEx, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing through November 13th, 2015. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Bye.